Hello and welcome to another day, another episode. It is wonderful to have you all here. And this is somehow the third episode this month we are having on malignancy in some way or the other. It's an interesting word, malignancy, and it's an interesting way of describe a disease. I think it's one of its kind where an adjective has been used to describe a group of diseases. And why is that? It's mainly because it is essentially describing a behavior that is evil, that was decidedly evil in nature because the dictionary defines malignant in something that produces death, that is persistent, that is evil in nature, that's passionately and relentlessly malevolent or evil. It has an intent of evil. It's aggressively malicious. Now, when we put all of these together, we understand why it has gone on to describe malignancy. But what is the main reason malignancy exists? What is malignant actually? And what is the hallmark of malignancy? Today, we are going to be discussing that in the biology of tumor metastasis, because at the end of it all, the hallmark of malignancy is metastasis. Without metastasis, really the tumors would not be persistent, would not be killing, and understanding metastasis is what is really behind everything else. And it could mean a lot of difference to how we go about treating and managing and understanding malignant tumors, basically. So today with us for this, we have Professor Dr. Lalitha J. Now, I have known her for a few years, actually quite a long time. She is a very kind, very unassuming, but highly knowledgeable person when you take the moment to stop and talk to her and discuss anything within oral pathology with her. And I was very happy when she decided to present this topic because I know she will do great justice to it. We also have with us Dr. Raghu Radhakrishnan, who's been with us a few times, and he's moderated the sessions and he's done an excellent job. He has I would say, an amazing knack of picking up the pertinent points, seeing that in depth, and summing it up, getting right to the crux of the matter. So these are the two people with us today, and I welcome them both. Very great friends. It's great to have them. And of course, I am your host, Dr. Mandana Donahue. With that, and with no further ado, I think we should go on to the session. So I will stop this share. Yes, and I will request Dr. Lalitha to please start her presentation. Welcome, Lalitha. Wonderful to have you here.
Thank you, Mandana. Thank you, Raghu. It's nice to be here. And a very warm good morning to everyone who's right now online. Right. Mandana, we start?
Yes, please. And a message for everyone out there while Dr. Lalitha is getting her share organized. If you have not already subscribed, please take the moment and hit that subscribe button so you can support this movement and continue enjoying all the good videos that we bring for you. Yes, now I will stop my video. Yes, Dr. Lalitha?
Yeah. Start sharing. Yes.
Yes. Yes, that's perfect. Thank you. Yeah, you can start. Yeah.
Good morning, everyone. I hope I'm audible to all of you. Right. Now, here we go. The first thing which we say in malignancy, we always know metastasis. And that is one word we never forget when we talk about malignancy. However much we try and treat the primary tumor, we are always concerned about the metastatic potential of the tumor which we are dealing with. Now, then that is the one thing which really concerns us. I'm sorry. Yes. So when we are talking about metastasis, we try to understand the process of metastasis, the drivers. What are the factors which drive this process? Right? The cell has gone rogue and then it stays rogue, but it also stays rogue silently for quite a long time and again starts manifesting in cell. And what are the factors which drive that? And trying to understand the drivers which drive these changes. The process. Metastasis is the hallmark of cancer. It's not due to the primary tumor, most of the patients are lost, but it is because of the dissemination of the disease to the distant organs, we lose our patients. And what is metastasis? Metastasis is the ability of the tumor cells in the primary tumor to move to distant sites, establish a niche for themselves there, colonize and proliferate, and again form new metastatic sites in different regions. Many a time, it's not only the primary tumor which is metastasized to a distant nation and formed a secondary primary tumor site there, but that again starts metastasizing. When we look at metastasis, it's a very simple, orderly sequence of steps. The cells which undergo a neoplastic change acquire certain changes which allow them to have the property to form a locally invading cell mass. And this locally invading cell mass is able to move across the stromal cells. They modify the stromal cells to allow them the movement across the established mature stroma which surrounds the primary tumor site. There are certain changes also which happen in these cells which allow them to intravasate into the blood vessels and into the lymphatic channels. They survive in the onslaught of the red blood cells which are just bombarding them, and also through the lymphatic channels. They survive in those circulatory channels, and then at the appropriate areas they extravasate. Extravasation is also possible, and they stay in a niche, and the environment, what we call the local environment for these cells, are modified even prior to the extravasation of these cells into these niches, and they stay dormant or latent for quite some time, and then again they start colonization. This may be a very simple way of putting it, but we must understand that it's just not as simple as this when we look at the whole process. So what happens here? So we have to look why these cells metastasize. Here the cell kinetics play a very important role. The cells acquire a cellular plasticity. We call this as the epithelial mesenchymal transition. This epithelial mesenchymal transition allows the cell to move across vast connective tissue stroma, and it also allows it to enter the bloodstream and colonize in a distant area. So why this happens? When a tumor cell starts proliferating, not all cells have the same type of genetic modifications. All of us are very aware of that. Once the cell divides in a very orderly sequence, the genetic material which is replicated is uniform and there is no change. But in a dysplastic cell, when the cell starts replicating, the genetic rearrangement takes place and we have different sets of clones. Some of these cells are able to proliferate locally. Some of them are able to elaborate certain signals which allow the modification of the underlying stroma. Some are able to even send out signals which allow new blood vessels to form to nourish the growing tumor mass, and also certain cell signals are able to travel to distant areas in the homing region and prepare the stroma for receiving these cells when and where they reach them. And we should also remember during this process of metastasis, not all the cells which start moving and enter into the bloodstream are able to home into the distant metastatic sites. The number of the disseminated cancer cells and the number of the circulating tumor cells are more when compared to the ones which reach their distant metastatic niches. So, having said this, we should now understand what we are going to do with this process, right? So, why is this metastasis still more important to us? The first is the genetic influence of these cells. The genes that are behind the tumor initiation and progression are the ones which facilitate the cells with the capacity to move and undergo an epithelial mesenchymal transition and also induce angiogenesis. The loss of the function of caspase 8 enables the tumor cell to infiltrate. And because of these cells evade the cell death, and these cells make up a particular portion of the tumor mass. And also the other growth factors, the transforming growth factor cascade, they are also very important in influencing the AMTs. So they promote these cells to switch between the mesenchymal traits and the cellular adhesion properties. See, what happens is these growth factors, we can't just brand many of them. So later when we are talking, we'll talk about the cell signaling. At one point in phase 1, they influence the cell and allow the cell to form a plasticity which allows it to move. But in the later stages, when they reach their metastatic niches, the same cell signaling pathway in their phase 2 make sure these cells which have reached the homing region are anchored in the metastatic sites and they proliferate to form solid tumors. So all these should be remembered. So we were talking about the growth factor beta, and these are also helpful. So again, what happens? The release of the cytokines such as the interleukins and the growth factors, the tumor necrosis factor alpha and the vascular endothelial growth factors. We must remember all these promote the alteration of the stromal, the connective tissue stroma adjacent to the tumor to facilitate the cells to move. And at the same time, when they reach their niches or their appropriate niches, they help them in reverting back to the near original phenotype and start forming colonies there. So all these should always be remembered. And the genetic influence is very, very important in this tumor cell. The other one which really facilitates the metastatic process will be the immune system. What happens here? The immune system is supposed to recognize the cells which are altered. When we say the cells are altered, whenever the genetic material changes, we should also know, and we are also aware that the altered genetic sequences elaborate proteins which are reminiscent of their embryonic protein forms and not of their adult mature forms. And this is where the immune system comes into play. The immune surveillance system is supposed to recognize those types of antigen which are not to be seen in a mature, fully functional, responsible cell and destroy those cells. But many a time it does not happen because many of these antigens have already been sequestrated or destroyed during the evolution of the cell to the particular phenotype, and the many a time these immune systems, they fail to recognize it. So the immune suppression on within the tumors are always mediated by a lot of chemokines and other cytokines, the tumor necrosis factor alpha, the tumor growth factor beta, the interleukin 1b, the interleukin 8, and so on and so forth. And then the list goes on, and all of us are aware of it. And these also ensure that the inflammatory signals are always maintained within the tumor microenvironment. And this also facilitates the extracellular remodeling of the stroma. And these are mostly influenced by the mast cells, the tumor cells associated macrophages, and the neutrophils. So all in all, when we look at this, the changes in the genetic composition influences the immune system in such a way that many a time they do not recognize the proteins that are expressed on the cell surfaces. And the next are the vascular, the angiogenesis inducing proteins, and the expression of them on the surfaces of the altered cells. The most important will be the vascular cell adhesion molecules, which are expressed on many of the clones of the tumor cells, which facilitate the adhesion of the tumor cells to the vascular endothelium and the intravasation of the tumor cells into the bloodstream. And extravasation is again facilitated by the integrins, which help in the opening up of the endothelial cells to allow for the extravasation of the tumor cells which are in circulation. And also we must remember these cells, these tumor cells which are in circulation, they form aggregates with platelets, and these help in removing these cells from immunosurveillance and protecting them as they move. And this, it may be a single cell or it may be a tumor cell embolus which are escaping the immune surveillance. The next one. So as you can see, what we have talked about, we are just seeing here that what happens when the disseminated cancer cells keep moving, and this is one of the only into the bone, right? When we talk about the bone, again, I will discuss it more in detail about that. Now, let us move on to the epithelial mesenchymal transition. So, we'll move on to the epithelial mesenchymal transition. Here, what do you mean by epithelial mesenchymal transition? The transition of the dysplastic cells to acquire the mesenchymal properties which were critical during the embryonic development and organogenesis is the one that drives the metastatic potential of the cell. So during this epithelial mesenchymal transitions, the epithelial cells lose their organization. There is no polarized organization of the cell here at that moment. And these cells are now migratory and invasive. When we say migratory and invasive, for example, in this squamous cell, they lose the shape of a squamous, complete mature squam, and they start reverting back to the embryonic cell form where you have got more spindling shape of the cell. And this helps the cells to intravasate, travel, and subsequently extravasate. And these secondary cells, when they reach the target organs, they again extravasate, and the cell signals that have been elaborated by this tumor at the very early stage. When I say early stage, even before we have diagnosed it as a malignant cell, these changes which are changed and which they have transported to the distant metastatic sites are able to prime the distant metastatic sites to receive these cells which extravasate. So these factors are secreted by the tumor cells, and the tumor cell stroma, which has produced changes even before we are able to visibly note those changes. These signaling pathways will include the Wnt pathways, the Notch pathways, the hedgehog pathways, STAT pathways, and the hippo signaling. So these are some very known pathways which have been elaborated, which are known to induce and allow for the modulation of the stroma to facilitate the epithelial mesenchymal transition. And post this, all these factors post the epithelial mesenchymal transition, once they reach their homing sites, there is a negative feedback loop which prevents the cells which have metastasized to again leave from the metastatic site and move to a distant site. So but for the fact, all of us are aware, yes, this happens, but every time one single pathway is elaborated, the next pathway automatically comes into play, and then the cell switches signals to move into the other pathway. So now, what are the stromal influences? What are the changes that the tumor cells make on the stroma which help in the switching of these cells to their more stem-like cell properties? So this we have to again look into. This is again facilitated mostly. What happens here is, I'm digressing here, I'm sorry. So let me just go back to the scene. When these cells have switched phenotypes and they have acquired a stem cell-like properties, they are more rapidly dividing and they have more tumor and they are able to produce populations which are more stable and which can form colonies in their distant areas. And how does this happen? These stromal cells, which are altered.
Dr. Lalitha?
Yeah?
Yeah, I think I will share your PowerPoint because your voice is breaking up. Yeah, I'll just share it for you and then you follow from there.
Yeah. I follow or I wait?
No, just 1 minute. I'll just share it.
Now, is that okay?
Yeah, and I'm giving you remote control.
Yeah.
So you can just forward the slides as you want.
Okay, fine.
Is that fine? Yeah, welcome.
Yeah, no issues. Thank you.
Okay, welcome.
Okay. So now what happened? We were talking about the stromal cells. At the leading edge of an invasive front, it is the stromal cells and which play 90% of the role of allowing these cells to move, and it is not the cells per se which are moving. This is one process which allows the more rapid movement of the cell. The cells are able to just stop. They need not stop cross-talking, but they just cross-talk with their stroma, and then they are able to move fast. And the second thing which we see here is the survival in the bloodstream. So up to 0.01% of the cells which intravasate into the bloodstream are able to reach their homing sites, right? Now, next. Dr. Mandana, I have the screen, but I'm not moving to the next slide, please. Yes, the drivers. Now, what are they? The drivers. The drivers are the different pathways which influence the conversion of the malignant cells, the neoplastic cells, as we call them, to move to the distant areas. And once they are able to do that, we are going to talk only about the Wnt pathway and the twist pathway here. We'll just make a brief mention as to how they move here. And before we talk about that, we will just touch upon the dormancy. What is cancer dormancy? Cancer dormancy is the cells are lying dormant. Why are they lying dormant? Why is it even after a period of 10 years, sometimes you have relapses? And there are some tumors where you have a relapse not in the primary subject, but when there is an organ transplant from the primary subject to a secondary subject, the metastatic tumor surfaces in the secondary subject and not in the primary subject, right? These are all examples of dormancy. So what does dormancy mean? The very fact what we call as dormancy is that many of the cells, the malignant cells, have the potential to lay quiescent for a long time. Dormancy is different from latency, right? The dormant cells are responsible for the tumor surfacing at a different age, and this is explained that the cells lie in a state of an arrested cell cycle. So they maintain themselves through they just maintain themselves with their stem cell like properties, and they do not allow themselves to be dividing cells. There has to be a complete new set of signals which has to be initiated for these dormant cells to again start dividing. And this is a very good concept, because when you're looking at cancer latency can also be, it cannot be equated with dormancy, because in the cancer latency, the cells are in a self-imposed quiescence, and you just inhibit one pathway, for example, the Wnt pathway, the proliferative population is just suppressed. And again, after that, sometimes when the signaling again is, when the cell signals are induced in the most proper way in their distant sites, they are able to start proliferating. And these are usually small cell clusters which escape normal diagnostic investigations, right? I mentioned this point very specifically here because when I'm concluding, I want to talk to you about a few lines about why I was stressing on this, right? Now we go on to the next one is the drivers. The understanding the drivers. Now we are going to talk about the Wnt pathway and the twist pathway. What is this Wnt pathway? This Wnt pathway and the hello, can you hear me? Hello.
Yes, yes.
Yeah, thank you. Yeah. So the Wnt pathway, the intratumoral heterogeneity in any primary tumor is always significantly associated with the progression. So when we are looking at it, the clonal organization and the dynamics which drive the Wnt pathway and allow it to metastasize is one of the most important pathways which we are looking at. This metastasis, which derive from the rare subpopulations using the Wnt pathway, is very, very important because there are two phases here. One is the phase 1 and the other one is the phase 2. This, along with the twist pathway, I don't know whether I'm able to put it very correctly to you. In the phase 1, both the signaling pathways of twist and the wing signaling allow for the movement of the cells by their mesenchymal capabilities to metastasize. And once they reach their homing site, the switching off of this, the first model, as we call it, the plasticity type 1 model, which drives the metastatic formation. And then the second model is once it undergoes and homes in the metastatic site, there is an irreversible AMT, and the cells lose their stem cell like properties, and they lose their plasticity, and they undergo a genetic change there, which allows them to home there and form the second primary tumor masses. And also the one thing which we have looked into is the natural killer cells, which are very, very important to prevent the destruction of these homing cells. And this is this is one of the factors where the natural killer cells do not recognize them when they are in their plastic type, it is in the type 1. And by the time they return to their type 2, their genetic material is altered and they are no longer an immune threat at that point of time. But further mutations can make them a threat to the immune system. Next. Next. So one of the most important distant metastatic niche where we look at metastasis, being a dental surgeon, we are more interested in looking at the metastatic sites in the bone. And bone is one of the common sites for metastasis. The thing with the metastatic event which happens in the bone is it leads to a lot of morbidity here. Any metastasis into the bone induces hypercalcemia, pathological fractures, and these in turn lead to a very poor prognosis. What happens here? Once these cells home into the bone, the whole environment in the bone marrow is changed. The first and the foremost thing is these cells are able to influence both the osteoblastic and the osteoclastic activities here. And they also influence the bone marrow progenitor cells wherever they are able to influence them. And one of the most important factors when these cells home into the bone cells are the induction of hypoxia and hypoxia-induced malignant phenotypes. These cells, which are the products of a hypoxia-induced microenvironment, are more stable to produce malignant phenotypes, which influence the destruction not only of the bone, but also influence the metabolic processes. One of the most important processes here, which they disrupt, is the metabolic syndrome. The most important here is the calcium levels are mostly altered for want of proper studies in the oral cavity. Many a time these they produce a sort of hypercalcemia, which is often overlooked as only as a metabolic syndrome, and it is not ever associated or ever thought of to look for a bone metastatic residue anywhere. And the next one here, when there is deposition of these disseminated cancer cells in the bone, will be the tumor associated macrophages, which are very important components whenever we are talking about metastasis. Now we also take into account the tumor associated macrophages, which are very, I wouldn't call it very, but they are the essential part in modifying the elaborating signals, which modify the stroma, which allows the circulating tumor cells to embed themselves. And next, these processes we are talking about multiple cells which go and lodge themselves in niches. But many a time we should also know that single cells or even a couple of cells can elaborate certain cell signals which cause a complete change in the metabolites. Not the metabolites, in the metabolism of the bone. And that is one of the reasons for hypercalcemia, which is often laid at the doorstep of the other tumors. Little do we know that the malignant cells which has metastasized to the bones, especially the mandible, are also capable of producing these all changes in the electrolytes and the other associated with the metabolic syndrome. With this, I think I don't have anything more to add right now. Dr. Mandana, you can tell me how we will go after this.
Yes, so I think we are back. Let's have Dr. Raghu also. And everyone on YouTube, if you have any questions, please share them. And yes, Dr. Raghu is with us. I think he can tell us something first, and then we'll hear what are the questions. Raghu, you're on mute.
Sorry, the only time that I am on mute is when the system mutes me.
Good one.
So thank you, Dr. Lalitha. That was a very comprehensive presentation of metastasis. You explained the biological drivers of metastasis, why the cells metastasize, changes in the genetic composition that alters the immune surveillance, angiogenesis, inducing proteins during metastasis. And also, I think you touched a little bit on the systems biology approach in understanding metastasis. So obviously, it was a very comprehensive presentation. So if I may just ask you, if you have any idea about the metastasis initiating cells? And sorry,
Yeah.
And also about the polyclonal metastasis?
Dr. Raghu, the metastatic initiating cells and the polyclonal metastatic cells, they influence these. But right now, since we were just talking only about the basics, I did not go in depth as to what were the drivers for the the changes. See, there are a lot of epigenetic changes which I could have mentioned here, which allows the cell to first start from the initial stage to the progression. So since we were just talking either only the basics, if I talk about those things, it will go very deep. I have just touched it very superficially. I have not elaborated a lot of points here because once I touch, that's why I made a mention here very specific. Or I have only expressed my views, and there is still a lot more in this. This is just the tip of the iceberg. You must understand. I perfectly understand. I have not gone in depth. I have just touched everything in every point. That's all.
No,
I have not made an
absolutely.
in-depth presentation or anything.
No, you have actually
I mean,
simplified it in few steps, how the
I-
local infiltration of the tumor cells take place into the adjacent tissue, the transendothelial migration of the cancer cells, which we know as intravasation, survival in the circulatory system, the extravasation. I mean, it
this
was
is
quite comprehensive. It was just not basic. Really
no,
appreciate.
Dr. Raghu. This is a very basic one, which all the oral pathology are expert. All our oral pathologists understand, and even if it is for the students, it is just what do you call a teaser for them to know what all can be done. But this is not the process. So when see, I can tell you just say when I say just epithelial mesenchymal adapt, I can tell you what are the signaling pathways which influence the primary tumor. So even before we see this plastic change, the cells are elaborating signals. It's not that they are not elaborating signals. Even before we see under the microscope and then say this is a very dysplastic epithelium, a severely dysplastic epithelium, the basement membrane has breached. But even before that, before ever we see this, a lot of signaling has taken place and a lot of modification has taken place in the stroma, and a lot of modification has taken place in the distal metastatic niches too. I have not talked about those things because right now this is not I don't have the time and space to talk about it. But it is there. We should be aware of this. When we say metastasis, we just can't say, all right, the cell has moved there, it has gone there, and metastasis is sat in that point. No, even before the cell starts going rogue, even one cell starts going rogue, that cell sends out a crosstalk signal to the adjacent cell, to the adjacent basement membrane, and that is why your integrins move, your basal lamina proteins change. Why do they change? See, we are not looking into that. See, as a student, yes, they should know that. But when we are talking in depth, we should know why it happened. That I have not spoken here. I've just told, yes, it happens, it happens. I suppose
Thank
it's
you.
not a,
Yes,
it's not
no,
a very in-depth
you're
one.
right. What has happened is I think it's going to be essentially the update series will have to take care of that now. The more in detail.
Yes, it has
Yes.
to. It's every single
Yes.
thing has to be talked about.
Dr. Lalita, your voice
Like
is
Dr. Raghu
breaking.
said, what's your
Let me see
like
what
Dr.
has
Raghu
been
said.
there on YouTube till your connection stabilizes a little bit. Let's see who's been there. So we have Dr. Nandini.
We have Dr. Sangeeta
Yeah, please.
Palaskar, Dr. Madan, and then Dr. Arpan Shah. Then we have Afreen, Dr. Ramachandran. My PG examiner is there, and Dr. Karpaka Salvi joined. Yeah, and so I've just sent a message if there are any questions to Dr. Lalita. I don't see any questions at the moment
And
from
besides the few who have commented, we are having 76 people who are watching. So there are many who are watching and not commented, so we don't really know they're there. Yes. So let's see, any questions came through?
there are no questions, Mandana.
Okay. So I guess we are done for the day. Yes.
Sure.
Yeah. Anything
it was
you
an
want to add? Yeah.
No, it was an excellent presentation. Really appreciate the effort that Dr. Lalita has put into this presentation. It was quite crisp, And I felt it was also quite comprehensive, although Dr. Lalitha maintains that it was very basic.
It's not a question of baiting Dr. Raghu. It is, you should have, like, I mean, it can go more. That's what I feel.
Yes.
Thank
It's
you.
always a matter of perspective, just how much in depth you want to go. But yeah, this was comprehensive when you think of the fundamentals. When you're learning the fundamentals, it was really very comprehensive. And the next level will have to be, I think, on the channel we have the the fundamentals, which is really for the PGs to build up their knowledge. And then although we have many of our colleagues also who are kind enough to attend every time and to attend to the discussion, but yeah, they're focused like that to go right from the basic. And then we will have the updates, which will then go on and take it the next step ahead. Yes, and I think that's it. Dr. Sangeeta Palaskar says, congratulations, Dr. Lalitha, great presentation. So yes, it is. Yep. And anything any of you want to say in general now about the way on the front of metastasis? I mean, aside of the talk, anything in addition you would want to add?
Mandana, I think basically laws of cell-to-cell adhesion, acquisition of motility, and the ability to digest through the basement membrane to enter the circulation is the beginning and the end of metastasis. So I think just to understand these three steps, it takes, I guess, several decades, because there are multiple driver genes, as Dr. Lalitha was explaining. There are a number of epigenetic factors that influence, and then you have got the evasion of the immune surveillance, which gives the cancer cells the necessary advantage that it has. And again, yes, it is the hallmark of metastasis, and once the tumor is in stage 4, it means that the most common cause of mortality is that as well, the metastasis. So I feel this is really pertinent in understanding how a tumor behaves and how the cell behaves. And as Dr. Lalitha was also mentioning, that these are not a linear cascade of events. These are overlapping processes where the cells assume a new phenotype, yet it will not shun its old behavioral habits. So, and it's also important that, like how you have the epithelial mesenchymal transition, and when it has entered the circulation and then the metastatic cell implants into a new organ, there is also a reverse process happening called the mesenchymal epithelial transition. Like how you have the EMT, you also have the MET. So I think these are all some of the things that we all have to be aware of when we learn about metastasis. And yes, today we had the tip of the iceberg. If Dr. Lalitha wishes to maintain it to be a very basic seminar, but it was a good start for your series of basic seminars. Thank you very much for this opportunity.
Thank you.
Yes.
Thank you, Dr.
Yes, it was an actually, you're most welcome. It's wonderful having both of you here. And I think there's the overall, we do a lot on cancer, especially within dentistry and within oral pathology, oral medicine, basically, because it's a big deal to us, especially in India. But we are really much more focused on prevention, on early diagnosis, and the very heart of what makes it malignant is really not getting as much of our focus sometimes, I feel, as the rest of it is. So this is a good thing to get people to realize that there's a lot of complexity there, and that also bears a lot of research, a lot of study, and maybe we can actually find an answer at that point. Because I think if we can break the cycle of metastasis, we have broken malignancy. And that would be something that we can hope that the next generation will look into. So thank you a lot,
you.
and thanks everybody also. I will
you,
have now
everyone.
one little share. Yes. So thank you, Dr. Lalitha, for this lovely presentation.
Dr. Raghu, and thank you for all of you who are there, who have listened to it. Thank you very much. Thank you.
Thank you.
Thank you.
Thank you. Yes. And don't go anywhere, Raghu. We have one for you also.
Oh, thank you very much.
Yes.
Thank you.
Hey, you're welcome. I actually sometimes feel like don't really need to give Raghu a certificate. He's so much a part of the channel now. I feel like it's almost like giving one of, like it's one of the members a certificate, but nonetheless, thank you. I know you had a meeting and you moved it. So thanks a lot for being here.
I don't know. Anytime.
Thank you. Thank you so much. And
thank you very much. Thank you, all of you.
Yes.
Thank you, Dr. Raghu. Thank you, Dr. Mandana.
Welcome. So coming to a few messages, and next week we have Dr. Anita Spadigam. We all, of course, know her. She is the HOD at Goa Dental College. I would say it is always a pleasure to listen to her. And she's going to talk on a very interesting topic, expressions of cell injury. What are the typical features you see when cells are injured? And this topic, in fact, was suggested by one of our viewers who said, and we would like to hear this. And Dr. Anita was kind enough to step forward and accept to do it. And next week, we are going to have her. So I hope you will all join us and watch that. That one, that is also somehow this month, we have a lot of fundamentals. So that is also going to be the fundamentals series. And I hope you will be there. And yes, with that, I think we are done for the day. And as usual, it is just the time to remind you that this is all about oral pathology, the subject we love, and it is for all of us. It's wonderful that so many of you come back every week. I know it's in the middle of a working day, and I know many of you watch it later in the day. But whenever you watch it, every single minute that you spend watching is supporting oral pathology. I'm highly aware of it. And I know you're also supporting me in this movement. I'm aware of that too. Thank you so much. And I hope to see you all again next week and the weeks after that. And well, have fun. Have a wonderful week. Thank you again.
Thank you.
Thank you.